From Microbe TV, this is Immune, episode number 68, recorded on May 8th, 2023. I'm Vincent Racaniello, and you're listening to the podcast about the body's defenders against disease. Joining me today from Ithaca, New York, Cindy Leifer.
Hi, welcome back, everybody. It is finally beautiful.
It's beautiful out, yeah.
Yeah.
And from Cleveland, Ohio, Steph Langle.
Hi there. Great to see you all. Yep, it's very nice here as well. Can't complain. It's very
Good
nice.
weather. Yep, spring seems to have sprung.
Yes, makes people happy.
And then when it gets hot, people will complain about the heat, but you know.
Yeah, I know. But it's a common thing, I think, humans. We have to, it's something we all experience. And we all like to complain. So it's kind of just something to talk about. You know?
And, we can complain about the weather, but you can't do anything about it. So,
no. So, right. It's kind of easy doesn't put the onus on anyone to figure out the problem. Yeah.
Today, we're going to teach you all about sin.
No.
Original antigenic sin, right, Cindy?
Yeah. So a lot of people have asked about this, and we've touched on it a couple of different times, but we thought maybe this time we'd give it a shot and see if we can kind of explain some concepts. And I thought the history behind this was really fascinating. So I pulled up a couple papers on this, and I wanted to go through a couple points, and we can just have a discussion here. It'd be great. Since these are review articles, they're not necessarily papers, although we do have one kind of at the end with a little bit of data that we might talk about.
Yeah.
So what is this original antigenic sin? I always wondered why it was called original antigenic sin. And I was really fascinated by the fact that the term was coined by Thomas Francis Jr. in the 1950s. And it was originally coined to describe patterns of antibody response to influenza vaccination. But what's really fascinating is Francis's father was a Presbyterian minister. And so Francis used theological terms to describe biological processes, which I thought was very fascinating. And so the idea was that first flu is forever. And so the very first time that you're exposed to an influenza virus is supposed to be your imprinting or your original exposure. And the idea then, they didn't know all of the immunological concepts, which we can touch on as we describe this a bit more. But the idea was that if you looked at older individuals, they had antibodies to earlier influenza exposures and young people did not. But then when they both got exposed to a new strain that was going around, turns out that the younger ones generated, quote unquote, a better antibody response than the older individuals who had pre-existing immunity to other strains of influenza. And the idea was, what if your prior exposure now made you less able to respond to these new things, which is interesting. But also, when Francis coined this term, he didn't really think of it in those negative terms. Mm-hmm. So he was thinking about it more like imprinting, which we can get to it. There's a lot of names that this original antigenic sin has been called, but imprinting is one of them. Mm-hmm. But his idea was that this could be positive or negative. And in fact, what he called things was like a blessing of induced immunity, which is, again, this kind of like biblical or theological terms. And his viewpoint was that if you were exposed to something before, you had induction of immunity that was protective. And I think it was just a bad choice of words to call it sin, because he really didn't imply originally that this was all a negative outcome, but that there could be interactions between antigens that you've been exposed to before and antigens you might be exposed to now. The key thing, though, in this idea of older individuals having antibodies around, and when they're exposed to newer thing, their antibody responses are lower, is that you're comparing something that's potentially broadening and re-stimulating to 0 to max in a child. So there's a lot of confounding things going on because older individuals respond differently to vaccines and exposures. So there's just a lot of really confusing, overlapping pieces of information that have been misconstrued over time that has people walking away with the impression that if you had a prior exposure to a vaccine, for example, and now you have an exposure to a different vaccine or the same vaccine again and again, what it's going to do is erode your ability to generate a new immune response. And that's the part that people have attached themselves to, but it's not really the original intent and not really the interpretation of the experiments that have been described over time.
Right. And the other nuance that would change your interpretation or the way that you thought about it is how distantly related is the influenza strain that you're now exposed to compared to the ones that you were originally exposed to. Yeah. That if they're more distantly related, you're more likely to have an immune response than if an antibody response than if they were closer together. And I'm sorry, I meant new immune response. If they were closer together, you're going to be recalling those memory B cells that saw an antigen very similar to that one. So you're right. There's no sin. It's just
it's memory,
the way that
right?
It's memory. It's memory.
It's memory.
Yeah. But it is funny in the flu field. And I think Scott Hensley was on TWiV and had some comments about this, but he's like, I know people bash it, but I actually really love the term original antigenic sin, and I'd like to keep using it. So I think it's fun too in the field to see who uses what terminology and for what reasons. But yes, and it applies not only to flu. We talk about this in enteric viruses. So norovirus has many different serotypes, and you will have this issue or this complexity of original antigenic sin for other viruses and probably bacteria, although that's not what I study.
But
I think, go ahead.
Cindy, do you know, so for influenza viruses, let's say your first exposure is an H1N1 influenza virus, right? If you then got an H3N2 where the hemagglutinin and the neuraminidase are really different, would that still give you the imprinted response? Do you know? I mean, how is it that there's always some cross-reactive epitopes that are playing this role? Is that the issue?
So I think the best data around that are if you remember, we had Gabriel Victoria on.
Yeah.
And he talked a little bit about this. And the idea was, I'm just trying to pull it up because I wrote some notes down. Yeah, so from his work, so remind the listeners if they hadn't listened to that. So he made a really cool mouse where they could track whether antibodies were being generated newly upon exposure to a new antigen or whether you were just recalling older ones that had already been class switched. So you generate an immune response, you'll refine those antibodies, and then you'll hold those memory B cells. And the gist of what his stuff showed was that it depended on exactly what you're saying, the distance of similarity between the second antigen and the first antigen. So the more similar they were, the more you relied on those memory B cells. And the more distant they were, the more you had a tendency to generate new B cells that could, with new specificities. Would you agree with that summary of that, Steph? That's what I remember.
Interpretation. So that an H3N2 challenge is not as similar as, the H1N1. And so that would be more able to recall new memory B cells compared to something that's in the H1 family. Although an H1N1 and H3N2 are still going to be more distantly, more similar than influenza B, which is a totally different lineage of influenza. So there's still cross-reactive epitopes for sure, but the ability to generate new memory B cells, is dependent on distance.
And what I liked about his work was that they said it's likely actually an evolutionary advantage because viruses tend to slowly evolve in general. Even with influenza, you've got antigenic shift that happens occasionally, but drift that happens over time. So you're more likely to encounter, and you guys tell me if I'm wrong, you're more likely to encounter a new virus that's pretty similar to what you already saw. Whereas occasionally there'll be a larger shift and it'll be much different. So therefore, it's very logical if we anthropomorphize the immune system to value prioritizing reactivation of memory B cells, which are very rapid in responding. And if they are at least somewhat overlapping in their recognition of some shared epitopes, that would provide, kind of that stopgap protection rather than trying to generate new B cells, which is slower to happen. Even if the latter one is going to give you some potential new specificities, in the short term, it doesn't really provide any value. However, when there's a shift, then you want to generate really new memory B cells. You don't want to rely on the really crappy ones that don't cross-react very well. So you're going to generate new ones. And so I think thinking about it from, what is it beneficial to the organism for survival, it's actually not a bad thing to try and call on the ones you already have if they're somewhat similar. So how does that play into all of this? Well, that's saying like if you now get exposed by vaccination or infection to something that's similar, you're more likely to recall those, memory cells that you already have to produce antibodies than to generate new ones. So if you got exposure now, it's going to look like this imprinting, right? So you're going to have a much more robust response to the things that you've seen before than the new ones when you're looking at that in the short term. I don't know if that makes a lot of sense.
Yes.
Yeah, it's just that I think it makes total sense, but there are some viruses that don't change substantially to become immune evasive, right?
Yeah.
So that's what's happening with influenza virus, as you just described.
Right.
But polio and measles, mumps, rubella, they are pretty stable. So I guess the other viruses were the selection pressure on the immune system. Or that happened a long time ago, obviously, to develop this imprinting, I guess. And so it must be it, because you cannot take every virus into account, right, when you're evolving.
Right. Because they're right. You have to have an ability to respond to just a huge plethora. And I guess with the viruses, Vincent, you were mentioning it, because they don't change that often, then it does become a game in terms of the vaccine strategy of making sure everyone has high enough titers, because those antibodies are effective against that virus.
Polio, the same vaccine we made in 1955, we're still using. Same with measles, 1960, and others. So, these are relatively new viruses compared to the evolution of the immune system, but it may be that when the immune system was evolving, and I don't know in what organism that would be, but maybe the pressure on it was something that changed so often that it made it become this way, right? That could be.
Right. Yeah, possibly. And, I think about those viruses that are zoonotic compared to those viruses that are not, and the adaptive ability then of those viruses that, like flu, because polio does not replicate in animals, correct, Vincent? There's no reservoir.
There's no non-human reservoir, no.
Right. So then it does, I think, bring this concept maybe into more focus is like, there's just so many recombination events. You have to deal with so many different distantly or non-distantly related flu viruses that, this is the way our immune system.
So that's a good point. Maybe, let's make the assumption that all human viruses were once zoonotic, right? They came from non-human animals. So maybe in those initial years after the spillover, there was a lot of antigenic variation, kind of what we're seeing with SARS-CoV-2. And if that was the beginning of some immune system, that could have applied the pressure. And then, as those zoonotic viruses became human viruses and many, many thousands of years became at peace with their host and didn't change as much, maybe then that part of the immune system is no longer needed, but there are other viruses that could still use it. So we're just speculating, but
we are. I'm sure there are people whose their favorite pathogen, they can email us, be like, no, this doesn't hold true for mine.
Sure.
But and it is interesting though when you think about vaccination for influenza, because it's the shift that causes the problem. And if your vaccines don't match, you're just not able, you don't have high enough titers or any memory B cells for that matter against that strain. And then that's the problem. So yeah, I don't know where I was going with that, but I was just thinking.
Right. So, and that's an interesting question, because I think a lot of the confusion around this original antigenic sin, for example, one thing that I've heard is people say, I don't want to get the yearly flu vaccine because that's just going to, not work very well, because you're just going to keep boosting to what I already have seen, and that it's not really protecting us very well. And I don't think I would agree with that, because what's about the flu vaccine is that the vaccine actually changes composition year to year. However, there are mixtures in those, right? So it's interesting to think about how might those mixtures influence the responses to the newer strains that you've added into the vaccine.
Right.
I don't know what you guys think about that.
Well, the flu, we give the flu vaccines a bad rap, but as Daniel Griffin points out, they do keep you out of the hospital, right?
Right. Yep. Which is the goal of really all vaccines when we think about it.
You may get flu, but it may be mild. And so there it is. It's fine. So maybe that imprinting is not bad, because those original antibodies may still protect you against hospitalization. Maybe they're broadly reactive in some way, right?
Right. So I think twofold. You're trying to boost, even if these are pre-existing antibodies that you, from a memory B cell you developed when you're a kid, you need to boost titers in these key populations like the elderly, who are the ones that are going to be in the hospital. So that's kind of the point of the vaccine, is boost your currently, whatever the match strain is. Now, if it's a non-match strain, we see more influenza-related deaths, but you hope that there's some cross-protective antibodies that are working for you, or they're not. And that's kind of the goal of a lot of efforts that the NIH has created, these consortiums to develop and I don't even like to use the term universal flu vaccine, because I don't necessarily think that's possible, but a super seasonal, something that is not just seasonal, that maybe you could extend it like 3 years or 5 years until maybe you need to change it again. And the concept is widely applicable to many different vaccines, viruses, and bacteria. We talk because we're virus focused, but thinking about the streptococcus, strep A conversation we had a couple episodes ago, hugely variable and a problem for vaccines. So
there's a good sentence from this paper, Cindy. It says, it's important not to generalize imprinting as a barrier to the generation of protective immunity, as is often seen in the literature. Right?
Yep.
So it's not a bad thing, as you've already said, not necessarily.
It really is that word sin that is, it's problematic for just a person who's generally talking about this. If you know the history, it's fine, but.
Right. Must be bad. Has to be horrible. Right. And I like, whoops, losing my earbud. I like the idea of this, back boosting. So they talk about back boosting, and what is that? It just refers to an increased antibody titers, like boosting towards antigens you've encountered previously. So all that, makes sense. That's what we do with vaccines. That's why we do that. But one of the things that they mention is that the increase in titers due to back boosting maintains the absolute titers towards past antigens at a higher level than the absolute titers towards new antigens. But you're still generating antibodies towards new antigens. The quality of an antibody is important, not just the quantity. And we don't always know what level of antibodies are protective.
Yeah.
Yeah, it is interesting. We're stuck with this. Somebody in one of the flu groups I'm in, I forget how he described it, but the unfortunate circumstance of the HAI assay, which is this hemagglutinin inhibition assay that was developed in the 1940s using, it's basically an adapted ELISA with red blood cells to see if you can block binding to sialic acid. And, the 1 to 40 titer correlates with protection, but it really boxes you into this one type of protection, which is neutralization by blocking binding. It doesn't bring into all the other different types of immune responses that could be protective that we've talked about before on past on the show.
Yeah, if it drops below, if the HAI drops below 1 to 40, they update the vaccine, right? That's the number they use. Yeah.
Right. I mean, and it doesn't consider the other things that are just harder to measure.
Yeah, sure.
Yeah, they're definitely harder to measure.
But what is also interesting is when you historically look at older individuals who, and I don't have it probably it's listed, but it's a pandemic year or it's an off year, and this one window of individuals are better protected because when they were children, they saw a very similar vaccine. So that's a benefit of imprinting, is that obviously you're going to be better protected if you see that again.
Exactly.
Right.
So it's not all bad. But it does call into question, mechanisms of how does it actually work? How does it happen that if you have already seen something similar, that when you see it again, you don't generate as good of an antibody response? So there's a couple of reasons why that would happen. One is that if you inject in an antigen or you get exposed to a virus, if you already have antibodies around at high enough concentration, they're going to bind to that antigen. And that might limit the ability of that antigen to go into new B cells to get presented in order to generate an effective immune response. So it's almost like you're getting exposed to a lower amount.
Right.
But that happens normally when you have antibodies on board that are protective, regardless of whether they are cross-reactive or not cross-reactive, right? So the whole idea of having enough antibody around, for example, in mucosal surfaces like IgA in the lung, is that when you get exposed, it's going to bind up the virus, and then you don't even have to make a new immune response. It's neutralized then. Right. So basically you're neutralizing the antigen before you can generate an effective response. But isn't that actually protection?
Right.
So this whole idea of you're not able to generate a new immune response kind of is saying that you already have antibodies that are around that are good enough.
Right. Yes. Yeah. And I think about this concept a lot because maternal antibodies so they're very beneficial. We know from many studies that they can protect infants when they cross the placenta or into breast milk, but they can interfere with the ability of the infant to generate their own endogenous responses, which is why we either do one of two things. We would just wait until 6 months when we think the majority of placentally transferred antibodies wane, or we would just give more doses to boost over whatever this threshold is. And so it's very similar concepts. We would expect maternal antibodies to bind up the antigen and prevent further processing by antigen presenting cells, or that there are some FC effector functions and that are actually inhibiting B cell signaling that could also contribute to this as well in the germinal center if those antibodies are around. So from an immunological mechanism perspective, it's very helpful to think through these things because it definitely impacted the way we thought about COVID vaccines. I never thought that the public would be talking about antigenic imprinting in the lay, which is great, but again, because it's nuanced, it can be very complicated to work through and to present like a clear message to the public.
Right. And that's the confusing part, right? Because it gets all jumbled together and the public is hearing original antigenic sin. And the only things that they can take away are, if sin. I got a vaccine, it's going to inhibit me from generating a protective response to a new variant, which isn't necessarily what it means. And that may or may not be happening. And, that kind of dovetails into the paper that you found, Vincent, from David Ho. Where they are actually looking at this with vaccines in naive or vaccinated individuals for COVID-19. And so I don't know if you want to go through that a little bit, but what the concern is different from what we've been talking about with influenza. Because influenza, you're thinking about changing the vaccine to match the current strain going around. And with COVID-19 vaccine, we had the original vaccine, and then we were using it for a really long time. And then we were still using it even when we knew that new variants have come out that are evading the antibodies that were generated from that. And so instead of moving to a new vaccine where you're better matched to the variants that are currently circulating, we just lumped it together with the original one. And we weren't really evaluating what the impact of that might be. So stepping back and thinking about it a little bit, if you've now taken the Omicron spike, you mix it in with the original spike, and you inject it into an individual who's had 3 doses of the original vaccine, what exactly is their immune response to the original component that's in that vaccine versus the new component that's in that vaccine? And what we've been talking about so far is that it might be that your immune system sees the old one, it likes that, binds really well to that. That might impact your ability to generate new memory B cells and new antibodies to the new antigen that's in the vaccine. Right? That's the concern.
Why would that happen? Aren't there not enough B cells? Aren't there enough naive B cells to accommodate the new antigen too?
Well, I wonder about that, because I guess it's just a numbers game. If you have low titers, then you wouldn't necessarily have antibodies around that would sop up all that antigen and make it disappear before you could generate immune response to it. But also, to me, it doesn't make a lot of logical sense, because if you've got the antibodies that preferentially bind up the original spike, they're going to bind to that. That's going to get cleared or endocytosed or whatever. That's going to leave more of the spikes around that the antibodies bound poorly to be now available to generate new antibodies and new memory B cells. I don't know what you think about that.
Yeah, I mean, I don't understand it mechanistically, but the data are pretty clear, right?
They are.
From this paper. If you give people bivalent, so you have people who've already been vaccinated, and you give them bivalent, they preferentially make antibodies to the original SARS-CoV-2, and they don't really get a broader response against the Omicron subvariants, right? But if you just give them a vaccine just with Omicron in it, then that does amplify those antibodies. But I just don't understand the mechanism there, because, those, as you said, the antibodies against the ancestral are going to be bound. So why don't you get new ones? Why don't you get new B cells engaged? I don't get it. Unless there's a lot of cross-reactivity that's somehow playing a role, right?
Which could be true.
Could be. The spikes are not totally different, right? They're really different, but there's still a lot of conserved regions. But
yeah, and that's where I think it goes back to the question with influenza, is like how diversion is needed. I mean, the amount of mutations that were picked up from the original spike to the new one there's quite a bit. It surprised people actually how many mutations were picked up. So, but I don't know what that means. Is it divergent enough, or they're still relatively similar in evolutionary timescale? So, I don't know. It's difficult, and there's only one figure here, so we don't get to really dive deep. But I thought, and I didn't pull it up for this episode, so somebody can email us. But I do think that's at least Paul B. Nash's group or somebody had published that people were able to develop antibodies against the BA4, BA5. So I don't know if they're just comparing different things or different populations.
Yeah, I don't think that this paper said they didn't generate any.
Right, just less than
just less.
Right. Okay. And I guess it goes back to the question we always talk about. Probably these people are all going to still be protected against severe disease.
Yeah, well, that's the thing in the end. Yes. In fact, all this is kind of moot because, in terms of severe disease, these people are all protected, whether you give them whichever one, right? So I don't know that the neutralization assays are telling us the whole story, just like the HAI is not, right?
Exactly. Right. And I think maybe it also bears to, we need to be probably subdividing, because I do think when you look at older individuals who are more susceptible for whatever reason, that the bivalent booster is likely going to provide protection. But if it's a young cohort, or at least a middle-aged cohort, I don't know if we'd see those differences.
So the problem is that the bivalent booster is not really boosting against the latest subvariants, which is what you would like it to do. So maybe we should just, so Ho's conclusion is take out the ancestral spike and just make it Moderna, just make it Omicron. But
yeah, and his rationale for that is he's insisting that that's going to compromise immunity and protection. And
we don't know that.
We don't know that.
No.
Yeah.
This is what it actually says in his paper. He quote from his paper is, "This analysis graphically demonstrates that inclusion of the ancestral spike in the bivalent vaccine precludes the broadening of neutralizing antibodies to BA.5, which was clearly evident in the breakthrough infection cases," which we can come back to. "Much of the world's population has been immunologically exposed to the ancestral spike through the vaccination or infection or both. The inclusion of this spike in the COVID-19 vaccines will continue to skew our antibody responses to what they have already seen and away from what we wish to elicit going forward. Therefore, based on observations made herein, we put forth a strong recommendation to remove the ancestral strain, the ancestral spike from the COVID-19 vaccines for the foreseeable future."
Two
Very
things.
strong.
Yeah, I was going to say two things. Wherever they publish this, I have no doubt they will make them tone that down. I do not think that will be as strong in the journal version. But, what do we think about making strong conclusions from breakthrough infections and not severe disease? These are two different mechanisms of protection.
Exactly.
Now, if I remember that we got 2 doses, and then the third dose really broadened the antibody to include Omicron, remember?
Right. Yes, I do. Yes, it was not a problem to bind.
So using ancestral is perfectly good at making it by somatic hypermutation and so forth. So the issue arises if you want to boost those antibodies, and then that's why they started including an Omicron component. And I'm not sure. I was never convinced that was going to help, but that's gotten us into this imprinting dialogue, I think. But I think it's premature to say take it out.
And probably that's why they haven't, right? Because I'm assuming everyone's looking at the same data, that they've not been convinced to do that. So, it's challenging when we don't. I mean, we need data with using the endpoints that we've been measuring it, which is severe disease.
The point here is that you put Omicron in, and but when you give this bivalent, you don't get higher titers against Omicron, yet they're still protected against severe disease, right? So maybe it doesn't matter.
Yeah, I mean, T cells are like, hey, we're here, just recognizing all of these. So
exactly.
Hello.
I mean, we did a T cell vaccine paper on TWiV Friday, and this is BioNTech has made a T cell vaccine consisting of epitopes from M, N, and ORF1, right?
Yeah.
And, when you combine that with spike in an mRNA formulation, in animals anyway, it does a little bit better than spike alone. But spike alone generated enough T cell epitopes to be protective. So it's just not clear that focusing on neutralizing antibodies is the way to go.
Yeah, I mean, it's hard, right? Because these things are all correlated. So neutralizing antibodies do correlate with protection against severe disease and death, but so do other things. So you use the thing that's easier to measure, but
exactly right. No, in the paper that we did, they were saying, non-neutralizing antibodies play a role clearly, via FC functions, and also T cells are playing a role. There are even some experiments where T cells can protect against, can reduce viral loads, right? Because they're eliminating virus-infected cells. So, but you're right, Steph, they're harder to measure. So we go for the neutralization. But Cindy, in addition to, the imprinting story has gotten a lot of momentum from influenza and now COVID. Are there any other vaccines where we worry about imprinting?
Well,
I don't think they talk about any other in this paper, right?
They don't.
I'm trying to think of the ones that are approved. I'm thinking of ones in development. Just because recently chatted about it, the norovirus vaccines. There's
right.
so many different serotypes that to pick, a G2 strain, but then it's very hard to keep up with the different amounts of types of serotypes you're going to get.
Right.
So that's an in development vaccine. But from a approved vaccine, rotavirus, I mean, that's a pediatric vaccine and it's a live attenuated, and there are different strains of rotavirus that can cause problems, but you don't see this issue. I mean, we have other issues with rotavirus vaccine.
Oh, Paul often mentioned that for the HPV vaccines, there's imprinting issues, right?
Oh,
yeah. It's a non-valent now, the newest HPV vaccine.
Yeah.
So do you get boosted with a different combination? Because I don't understand how that would work, because if it's a 2-dose vaccine and you get the non-valent twice, how would imprinting affect that? Is it because you have had prior HPV exposures?
That's what Buffett said. Yes, it's what you were exposed to previously. Yeah. Yep.
I don't know the prevalence of that one in the early aged populations. Is that really common?
Don't know. I just know he mentioned it on a TWiV that was that you did see imprinting, but I'm not sure if it has any consequence for disease, right?
I mean, we know that's hard. Yeah, there are more pathogenic strains of HPV
right.
16 and 18.
Yeah.
But then it's expanding now to as we had brought down incidence of 16 and 18, now we're seeing HPV 33. And so then but I'd have to, for a DNA virus, I'd have to think about that more. I mean, does it happen? Sure, imprinting happens, I think, for all pathogens, but does it affect the vaccine? I'd have to think on the HPV question.
I mean, it's only going to be an issue where you have multiple serotypes or antigenic variation, right?
Right. Mm-hmm. Sorry, yes, that correction is needed, right? It's not for all of them if they're not changing.
Wow. So we didn't actually say which paper we were kind of dissing a little bit with this, because we said when it eventually becomes published. So this is from David Ho's lab. It is on bioRxiv.
Yeah.
So that's why we were questioning a little bit, because it hadn't quite come out yet in its full form.
And the data is good in the sense that it's there. It's definitely clearly telling us something.
Yeah.
I think that just last paragraph might have to be, or whatever that paragraph is, might have to be toned down a bit.
Yeah. So the paper that I pulled out originally to start talking about this is a paper that just came out May 9th. Tomorrow. It comes out tomorrow in Immunity, which means it's already been out for a little bit. But it's called Immunological Imprinting, Understanding COVID-19 by Marios Kousakos and Ali El-Bedi. Yeah.
Cool.
And where I thought it was interesting to mention a little bit. So what the Hope paper is doing is making extrapolations from, like you said, HAI data or neutralization data, and trying to make epidemiologic and health practice suggestions based on those data. And there was an interesting little part in the other paper, in El-Bedi's paper, about extrapolating and how we need to be careful about that. And so it's saying like numerous factors have to be considered if you try to extrapolate the immunologic findings. So, virus neutralization, if you're not looking at clinical outcomes per se. And so I have to read this because I don't know if I'd summarize it correctly without reading it out loud. So bear with me. This is, for example, analysis of one group of individuals exposed to strain A and then to strain B will typically reveal higher antibodies towards A than to B. That makes sense. This is evidence of antigenic seniority. So they call it seniority.
Another way to say it.
Right?
Yep.
Right? But does not constitute evidence that prior exposure to A has any negative effect on antibody responses to B. So to determine whether such interactions exist, the analysis needs to include a group not exposed to A. As a comparator is frequently missing from the literature. And this too, and it becomes almost impossible to do because pretty much everybody's been exposed to COVID-19 at this point, either by infection or vaccination. So it's very hard to say, what happens if you just see the Omicron spike that's in the vaccine alone, because we're not even injecting it that way, right? Alone without having seen A. So you have no comparator to try and figure out what the immune response to that antigen alone would be.
Yeah,
so we have to rely on animal studies to do that.
Right.
But they say when such a group is included, there are 3 possible scenarios that can happen regarding the antibodies against the second thing. First, the individuals may have the same antibody titers as the people who see B alone. And this would indicate that there's no effective prior exposure to A. The second is the individuals might have higher titers against B than people who only see B, which would indicate that there's actually a positive effect of prior exposure. And then the third possibility is that the individuals might have lower titers against B than the B-only people, and this would indicate negative effects. But by inferring both, epidemiologic outcomes on an individual or a population basis of, protecting you from severe disease or death, or just, in the population, extrapolating that from an in vitro serological titer neutralization is dangerous, especially when you don't have these comparators. So I think that was really valuable to think about, because we may see something that's lower, but does it actually mean anything? And I don't know that we'll ever know the answer to that because we don't have those comparators to do that actual study.
I think, yes, that was I'm glad you read that, because I think it was really clear and made sense of what we're talking about. If you could find those small pockets of people who did not get vaccinated, who did not become infected with the wild type, but did get infected with Omicron, I'm sure they exist.
Yeah.
Maybe small, but that would be the comparator group in this scenario.
Right.
So
and
you're
maybe
right.
the serum samples are out there and maybe somebody needs to do that study.
Probably banked somewhere, I'm sure, in somebody's biobank. They do. But it's either no effect, it's a blessing, actually, or
it's a sin.
Right.
Blessings and sins.
Right.
Yeah, they have a paragraph here where they go through the COVID issues. And they say, in the end, though, any exposure still
is protective.
Is protective against severe disease.
Yep.
So, we're talking about this, but it may not matter. I mean, the mechanism is interesting. I agree, but I think for David to say, take it out, David Ho, is a bit premature.
I would agree with that.
But, that will make it in the press, right? And the press will say, experts call for changing the vaccine.
When did this drop on bioRxiv? May 4th, so a couple days ago.
We can Google David Ho's name and see if anyone quoted it. But there's a lot of other public health things going on right now. So, in terms of unique to America. So I'm just wondering if the press isn't focused as much on bioRxiv as they once were, maybe a year ago.
Maybe.
But it's probably worth mentioning, though, that both the WHO and the US are basically saying the pandemic is over at this point.
Yeah, that's interesting.
How do we feel about that?
It's funny because I forget who I heard this from, but, defining a pandemic is really challenging.
Yeah.
HIV could still be considered.
It is.
I mean, it is. It's still a pandemic. So it's a combination of the acute problem of the virus still causing issues.
So it's a great, we just had a textbook meeting last week.
Yeah.
And I said, we need to come up with a definition of pandemic.
Oh boy.
And I submitted one. Okay.
What was yours? Can you read it to us?
Yes. Where did I think I can recall it. I think I can recall it. So pandemic is a worldwide outbreak that infects many people in many countries and affects and alters the way you live, work, and play.
Mm.
So that last part, I don't know where I got it from. So I think it was a review article. So I think Theodora said, well, we're in an HIV pandemic. How does that affect how you live, work, or play? And I said, well, it could never get grants because it all went to HIV/AIDS.
So that did affect you.
So it affected me indirectly, right? But I would say we are still in a HIV/AIDS pandemic because it is spreading. It's very difficult to control spread. You could treat people with antivirals, right? But it's still spreading. In many countries, people don't get antivirals. But we've forgotten about it, essentially. That's why people don't think.
Yeah. So here's kind of an opposing view.
Yeah.
If you think about outbreak, an outbreak is more cases than expected.
Right.
Over a certain time period in a certain location. And so I think as you begin to expect a certain number of cases, and you can predict it on a seasonal basis or on a yearly basis, is that still an outbreak or, an epidemic is sort of, larger outbreak, and then a pandemic is a larger epidemic.
Yeah.
So it's an interesting question because if cancer has a predictable number of people per year that get it, that's not really an outbreak or an epidemic. But if there's a jump in the number of cases of a particular type of cancer in a location, then we start to go, oh, hey, what's going on there? That's unusual. Right. So with HIV, you have a certain number of expected cases all the time. It's still spreading. Is that really an outbreak or an epidemic? Or is that just the new normal? And so with SARS-CoV-2, are we reaching a new normal, or should this still be considered an epidemic or a pandemic? It's an interesting thought question.
Yeah.
Now, there are many viruses that infect many people every year, right?
Right.
Like noroviruses and rhinoviruses and even seasonal flu. We don't call them pandemics. But
what about the concept of seronegativity, that the naive population? Did that get talked about when you met with your book?
So interestingly, the one I read a review article on defining a pandemic, and they said the original definition of a pandemic has nothing about population immunity, virulence, or transmissibility. It has that was left out. It was all about a big outbreak, basically.
Like what Cindy's saying, just an increase in the number of expected infections.
But I would say it has to
correlate.
It has to have some kind of doom component, right? That's right, because you get a rhinovirus outbreak, you get a norovirus outbreak every year. We don't call it a pandemic because why? I guess we can handle it. We can give people intravenous fluids and mostly save their lives, right?
Right.
But with this one, as an example, COVID, we have no way of stopping it. We can't protect people. There's no antivirals. You can't give them anything. You give them high flow oxygen at the end, but many of them die. So I think that's the scary part, that you can't do anything. And so the WHO wants countries to be ready for that.
Right.
So that's how I look at it. I don't even think it's a virological definition. It's just something bad is going to happen, folks, and you should be ready. or as I said to the authors, I can't define pandemic, but I know one when I see it. Right? That's really it, because sometimes you can't pigeonhole yourself to a 2-sentence definition. So, like the Supreme Court judge said years ago. Anyway, It's so interesting.
It is.
By the way, Maria von Kerkhove, who's sits at the left-hand side of Tedros at WHO meetings, right? That's her name? Maria von Kerkhove.
I know her face, yes.
Anyway, I'm going to have her on TWiV next week in Switzerland.
Oh, fantastic.
That's great.
She's the technical lead of the COVID-19 response and the head of emerging diseases and zoonoses at WHO.
Awesome. Cool. So ask her what a pandemic is.
I'm going to ask her what a pandemic is. I'm going to also have Kanta Subaro on.
Oh, excellent.
Who's the head of a WHO collaborating center in Australia. So that'll be fun.
And a flu person. So, I mean.
Yes, we got both of the big ones, right?
Yeah. Is this at the coronavirus?
It is, where you're not going to be, unfortunately.
No. International travel was not in the books for me.
No, May, I know. So. But if you had come, I would have had you come on with me. You could have been a guest co-host for TWiV.
I would have loved that.
You could do it next time.
So that now those meetings, because I went to one, you saw me in 2018 before.
I did.
Very small. We were in Kansas City, mostly like animal coronaviruses were getting talked about. But that was every 3 years. And then, of course, it was delayed. Do you think that they're going to have it more frequently than that? Or I guess you can tell us after you go.
I'll find out. But there are 340 people attending. Is that about what there was back then?
Yeah. I think that's interesting because I wonder if they kept it smaller on purpose, because it is nice when they're smaller.
Yeah.
Maybe it's a little smaller than that, but yeah. Cool.
Anyway, I don't know. I'll ask the organizer is Volker Thiel.
Yeah.
I'll ask him what their plan is, but it's hard to, you can't tell from past because we've skipped a few years, right?
Yeah.
Because of the pandemic, but
it
should be interesting.
That's funny that a pandemic got in the way of a coronavirus meeting.
Hey, a pandemic canceled my virology course, 3 years ago. I thought that was the weirdest thing, right?
Yep.
Actually, it didn't cancel it. We had to do it by Zoom, right?
By Zoom,
yeah.
Gosh.
Wild.
Things have been coming up. My memories say, 3 years ago, everything shut down. Everyone was at home.
You're like, listen, social media, my brain is purposely block that out. I don't need to be reminded.
Yeah, you're
this
right.
is when you baked your sourdough bread. Didn't everybody do that?
But you had to get a starter though, right?
Yeah.
I couldn't believe it was happening when it was happening. I said, no, this can't be. Can't we deal with this? Aren't we better? Don't we have the knowledge to be able to deal with this? Apparently not.
Well, what I'm worried about is whether we're actually in worse position.
I'm normally an optimist, which you guys know, but I agree, Cindy. I am very concerned that we are worse off now, public health wise, than we were.
So why do you think? Tell me why you think that is. I agree with you, but why do you?
I think that I am concerned about the CDC leadership. I am concerned about the public perception of vaccines and therapeutics, and I think you just would have so much pushback based on the people I interact with. I'm concerned.
Yeah, I'm nervous that if something like this happened again and we were able to generate a vaccine really quickly and we know exactly what works with wearing masks and everything, people are going to be like, nope, not doing it.
Yeah, I think people are
been there, done that,
or they distrust.
I agree.
It didn't make any difference then. It's not going to make any difference now. I'm not going to do it. And they're not going to do it based on science, and we're going to end up in a really big situation.
Yeah, I agree. And I think the underlying reason is the politicization of science and public health. Right? Some politicians decided they would be anti-vax, anti-mask in the name of independence and choice, and they got their voters to vote for them, and that's all they care about.
Yep.
Right.
And I do think it's just a flaw of our species that we think very myopically. So like if it didn't affect, if I'm not, I mean, some people did get affected. They killed, it killed their family members. But I think we only have an experiment of one. We don't have the comparison if we didn't have vaccines and didn't try to use masks, that it would have been worse. But we can never do that. And it's hard for people to assess risk. It's just it's challenging.
And now we have a presidential candidate who is an anti-vaxxer.
I know.
This is sad as anything. And people just don't realize how sad this is, that the stalwart of saving people's lives, public health, is going to be a campaign issue.
I think that is it's going to be. So we have to keep doing this. And even if we don't get as many listeners as other people.
We will. I mean, I'm very glad that the two of you are here and you guys never complain and all the other hosts never complain. And I want all our listeners to realize that everybody does this without pushing. Nobody has to be pushed to do this because it's really important.
Yeah.
because public health is meant to save your life. And for you to think otherwise is, is just not right.
Yeah. No, we do have, I mean, to think about, it's interesting because maybe I should eat my own words a little bit, because we do have a new vaccine, and I'm going to be very interested to see how the uptake of that goes, and particularly immunocompromised populations. Do we want to touch on this new vaccine that we're going to have shortly?
Yeah, we could. Yeah.
That's up to you.
It's up to
You've got a little bit of time.
Yeah, we do.
Just mention it.
Tell us about it.
So respiratory syncytial virus is a vaccine whose development has been problematic historically. There was a vaccine that did cause worse disease, and this was a long time ago, before we had the advances that we do now to understand why, or how to make them better. And so people wouldn't touch RSV vaccine development for a very long time because of this issue. Respiratory syncytial virus is a respiratory virus that mostly is going to be killing babies, young babies, neonates, and the elderly. And it really compromises your air sacs. It fills them with fluid, and you basically drown in your own lung fluid. It's terrible. So, but advances in the structural basis of this virus have allowed for a vaccine that has demonstrated protection, and they have approved it over 65. And I think they will soon approve it in pregnant women, so that they can passively transfer it to their babies who wouldn't be able to otherwise get vaccinated. Mm-hmm. And, Vincent, maybe you want to, I don't know because you got to talk to Jason McClellan, him and Barney Graham at the NIH were really able to solve this issue of pre versus post fusion conformations of the F protein of this virus. And that has led to this vaccine.
Yeah, it's just brilliant. The fusion protein, when it interacts with a cell, undergoes a conformational change, and it's really different. And those epitopes are not the right ones for blocking infection. So you want to keep it in the conformation before it undergoes that change. And turns out adding a couple of prolines at the right place in the protein does it. It keeps it in the pre-fusion. So now you have the right epitopes, and that's what was the key to the spike SARS-CoV-2 vaccines, and now RS, which I think it was tried first in the RS vaccines, right, before SARS-CoV-2 even emerged. Right. So this is is this Pfizer's vaccine, the RS? Who's making this? Do you remember?
I think GSK is it's called Advair.
Yeah, right. And I think Pfizer also has one that may be some news coming up very shortly. Daniel hinted.
Yeah. Did he? Oh, good.
They have, which I don't know if it's an mRNA vaccine, but the one by GSK is a protein vaccine. It's a purified protein, F protein, and it's adjuvanted and injected. And I'm going to get it. Why not?
Yes. And so I think it goes back to when you make a whole population get a vaccine because of the global issue, that's when things get very problematic, and people then antagonize, use whatever their motives are to antagonize public health. But I think for this, because it's very targeted to a population that needs it, when I brought it up to people who otherwise are vaccine skeptics, I think a couple of them who are older are like, oh, I'll get it because, they're susceptible to, so it's interesting. I don't, I do wonder if it's the comparison between the like your job or something making you get something compared to something that's elective. I don't know.
Which shocks me because, if they're making you do it, there's probably a really good reason why we need this, right?
Yeah.
We don't mandate things just for the fun of it, usually.
Who wants to get death threats, right? Like, I'd rather not have to mandate anything.
No.
Okay, so here's the package insert for Arexv.
Okay. That's such a silly name, but okay.
Approved for use in individuals 60 years of age or older. So they did a phase 3, and that's the basis for the approval. Protein is made in Chinese hamster ovary cells, CHO cells. So it is produced in this these cells. It's secreted into the medium, and then the protein is purified by several chromatographic and filtration steps. Good old-fashioned biochemistry.
Yeah.
And then it's mixed with an adjuvant, which is monophospholipid A from salmonella. And you know people are going to say, aha, they're injecting salmonella into me. Right?
No.
But this is there's two adjuvants. It's the monophospholipid A and a saponin purified from a plant combined in a liposomal formulation. Interesting. So then that's injected into you, and there you go.
Make antibodies against the pre-fusion F protein.
And the vial stoppers and the vials are not made of latex.
Oh,
so if you have a latex allergy, it's not a problem.
You're good. No eggs. Good for egg allergies. Good for latex allergies.
Arexv. I wonder where that came from. Arexv.
They have groups of people that's their job to make up something that doesn't sound like anything in any language.
Oh, is that the goal? So that it doesn't
That's
sound
why
like
it always has so many X's and Y's and unusual like BK type of like letter combinations that don't normally come up.
Oh, okay. That does make sense. Because you wouldn't want it to be associated with someone's culture or language. Okay.
Right. And then I just saw that, so the phase 3 trial that you're talking about had a good number of participants. So it's 12,500 received Arexv and 12,500 received placebo. And the vaccine significantly reduced the risk of developing RSV-associated, LRTD. I don't know what that is.
Lower respiratory tract disease.
Yeah. Lower respiratory tract disease. Yes, thank you. By 82.6%, and reduced the risk of developing severe RSV-associated LRTD by 94.1%. Those are pretty good.
It's pretty good
Statistics
good numbers,
for vaccines. Yeah.
Oh, wouldn't that be amazing if we could just squash RSV season? That wasn't even a thing.
Good.
That would be great. Yeah.
So, Steph, would you predict that any antigenic variants would arise?
I have to say, I don't know a lot about RSV replication.
Okay.
So maybe would lean on you for that. But RSV, it's not jumping into other animals. It's pretty stable. So, you will have mutations that avoid, right? Just that's how viruses work. But whether that would evade or I don't know. Yeah, this may be able to compromise the virus enough that it doesn't mutate away, but it also I don't know. It's hard. This is predicting evolution.
So there are there are two antigenic groups already, right? A and B. And within them, there's some variation. I don't know the vaccine, which one it's produced from. That's a good question. Right?
Yeah.
Let's see if the package insert says that. No, it doesn't. Maybe they're not that. I don't know. So I'm going to plead ignorance here.
Okay. Yeah. It's hard to say.
But I don't think that the vaccine is going to do anything that wouldn't have happened already by infection, right? A lot of people have been infected. There's plenty of pressure from that.
It's a lot of immune pressure.
I see what you're saying. Right.
Yep.
Like we shouldn't expect something crazy to happen. Yeah.
Hey, cool. Very good. All right. You want to talk about this art thing, Cindy, or whoever?
Oh, yeah.
That's cool.
There was one last thing I wanted to mention. I just happened to come across this and I thought it was really neat. So there is an art competition. Let me open up the link. It just happened. So it's at the La Jolla Institute. And it's a competition for middle school and high school students to draw something immunology related. Wow. And I just thought it was really neat. So we'll put the link in the show notes and you can go and you can look at these drawings are just amazing. Different kinds of art pieces that depict something related to immunology. So they have a junior immunologist category for ages 10 to 13. The first place winner was Isa Esnani. She's High Tech Explorer Point Loma. And hers is called Healthy Heart, a tribute to Tio Pablo. And it's this really pretty picture of a heart with flowers. It's really neat. And then they have a senior immunology category for ages 14 to 18, and first place, I can't view all the winning pieces, but first place was Judy Kang, Westview High School. Is this hers? No,
yeah, you're right. The 2023 winning pieces
are not quite
link
there
like the others. Yeah.
Yeah. But they do have the links to the other winning pieces from previous years. And they're just, some are very detailed. Some are really just abstract.
Wow.
But I find it really interesting because one is, for example, milestones in vaccine history. So these students are thinking about, vaccines, the pandemic.
Yeah.
Some are, this one from 2022, I actually think is cool. Third place winner for importance of public health policies during the pandemic. And you can see like someone coughing and their red particles coming out of their mouth and somebody wearing a mask. But it's interesting to just view what these students are thinking about and expressing in their art. Like, I'm not a particularly big art person, but just like if they've experienced cancer or they lived through the pandemic or something and how they're choosing to express those in art, I think is really telling about how the next generation is growing up and thinking about immunology and just infectious diseases.
This one you just described is great. It's black and white, and then the droplets coming out of her mouth are all red. Isn't that great?
Yeah, and the other people walking down the street who don't have masks all have red sprays, and the ones who have masks don't have red sprays.
It's really good. I like it.
And the drawing is, I'm impressed because I couldn't even draw anything that looked like a building and you'd be able to know it was a building. So the fact that they can do all that is amazing. There's probably stuff in the windows that, it seems very symbolic, right?
Yeah.
It's very cool. Yep.
Yeah.
Cool. I like it. I like it very much. These kinds of competitions are great when you get engaged people and they get creative and really nice. Cool. It's organized by the La Jolla Institute for Immunology, right?
Yep.
Great, where our friends Shane and Alessandra work, right? And Daniela, all of whom have been on TWiV. All right, so we will put links to that in the show notes so you can see them. And if you want to know where their show notes are, they're at microbe.tv/immune. And that is episode number 68. You can find the show notes there, microbe.tv/immune. If you want to send us a question or a comment, you want to fix something we said that you don't think is right, feel free, immune@microbe.tv. If you enjoy our work, consider supporting us. We would love to have your financial support if you like what we do. Microbe.tv/contribute. Cindy Leifer, Cornell University, Cindy Leifer on Twitter. Thanks, Cindy.
Thank you.
Steph Langle is at Case Western Reserve University. Stephanie Langle on Twitter. Thank you, Steph.
Yeah, thanks. This was fun.
I am Vincent Racaniello. You can find me at virology.ws. Music on Immune is by Steve Neal. Thanks for listening to Immune, the podcast that's infectious. We'll be back next month.